Okay, everyone. So we are in our next segment, and this is going to be a multi-chapter segment. But like I've been telling you guys, I think for the last couple of videos, is that these next several chapters will be just based on the PowerPoint. So I would highly encourage you guys to take a look at the textbook as well as the PowerPoint title. And whatever we mainly focus on in the PowerPoint is what you guys should be focusing on when you're studying for the next exam. So without further ado, let's just get started. I don't want this to be a 2-hour video, so we're gonna get on with it. And so the last chapter we've kind of discussed was let's see, it was chapters 7 and 8. So we were talking about the regulation as well as the analytics of how do we know that the protein that we have is a protein, or how do we know that the DNA we have is a DNA? And that's through the Western blot, Northern blotting techniques, as well as the concurrent addition of the cell fractionation or the gel electrophoresis or even the column chromatography for specifically chapter 8. That's what I want you guys to focus on. Now, in terms of regulations, we're going to take a step outside of the nucleus now. Now we have an understanding of how DNA to RNA, RNA to protein is transcribed and translated, and the regulation that encompasses that, whether it's the lac operon or the trip operon or any other regulatory processes. We have an idea of how that works now. And now we're going to get into the other aspect of the cell. Mainly, we're going to start delving into the cellular trafficking and cellular transport. And there is a great YouTube series that they just started posting uh, on our textbook. And so I highly encourage you guys to kind of look it up. It's like, I forgot the name. I'll post it in an announcement very soon, but it's a last minute lecture. And the idea is that they summarize the whole entire chapter in about 30 to 50 minutes, depending on how detailed the chapter is. And it's a great format where it's pretty much two or three people having conversations with the topic at hand. Now, fair warning, if you guys are going to look up this YouTube series, it is a great, I've listened to a couple of them. It is a great way to kind of get an overall view of it. I recommend having your PowerPoint or textbook open while they're talking because they go through the concept very fast. But it does seem like it is AI scripted, or if anything, it's AI speaking. And so just forewarning you guys, it's great. It touches on a lot of things in a very simple, more advanced than what I will speak normally, but it is very simple to kind of follow along. So again, it's last minute lecture. Just type it on YouTube, Albert's lectures, Bruce Cell Biology Upper Division in YouTube, and it should have a image of our textbook like this right here. Okay. I will be showing to you guys in the lecture. So if you guys are coming to lecture, I will show that to you guys. But yeah, there we go. Anyways, enough of that. Let's get started. So we're going to talk about membranes. Now, you guys have an idea of what membranes are. Membranes are phospholipid bilayers. They're comprised of fatty acids and a glycerol cap with some sort of altercation or alteration of the third carbon on the glycerol chain. Now, for us, it's important that we kind of understand that we're stepping away from GenBio and we're going into the more advanced things, which is what happens on a phospholipid itself. Now, we're used to seeing this as, okay, here's my phosphate head, and here's my two fatty acid tails. So that is what we generally teach you guys. As starting out, you guys have an idea that, okay, well, this is going to have, an amphiphilic behavior where one side is polar, the other side is non-polar, one side loves water, the other side doesn't like water, and that's going to cause some sort of interaction and behavior, which is absolutely true. You're going to have one side that is going to, be hydrophobic. But one thing that we need to understand about the membrane is that number one, the phosphate heads can have attachments to it. So again, you're just going to have a glycerol cap where it's three carbons, right? Two of the carbons will have fatty acids attached to it. And then this third carbon will have a phosphate group, but then something else can attach to this phosphate group. Now, depending on what's being attached to that, will depend on where it is found, if it's going to be a transport vesicle, is it found, in a particular cell type, all of that. So just keep that in mind. Now, on top of the bilayer that we've taught you guys already, one thing to note is that yes, there are cholesterols found inside. So these green little bits right here are your cholesterol bits, and they add into the fluidity of the membrane. Remember, everything is constantly in motion. All these proteins are not staying put like you see on the screen. They're actually flowing back and forth like a bowl of soup. Things are moving back and forth. They're never stationary. They might be stationary for a couple microseconds or even milliseconds, depending on the need, but it will still be moving. Okay. So it's always in movement and it's a two-dimensional fluid. Okay. So this is what I mean by the addition to the phosphate head. So we do see the two fatty acid tails. Now these are your omega-6, omega-9s, omega-12s. You have your glycerol cap right here. This is your glycerol cap. So these are the two monomeric forms of lipids as we review from chapter 2. But you also have this phospholipid head, phosphate head, and then you can attach certain things to that phosphate head to give it a difference in behavior, functional difference, etc. So what we have right here is a phosphocholine or phosphatidylcholine, sorry, where we're adding a choline group to this phospholipid. Yes. And the choline group is literally adding another nitrogen group right here. It's not an amine group because we're replacing all the H's with methane groups or methyl groups, but it is going to cause a positive charge on top of this phosphate head. Now, you guys know phosphate is naturally a negative charge. So with a choline here, it kind of neutralizes the head out in terms of the electrostatic charges. Okay. Now, there's different types of classifications that we have on our phospholipids, for instance. So there's going to be four major phospholipids that is found in the plasma membrane. It's not just phosphate group, but it is going to be phosphatidyl ethanolamine, phosphatidylserine, choline, and then sphingomyelin and sphingosine. Now, sphingomyelin is made from sphingosine. Okay, so sphingosine again is just a precursor. It's involved in other processes inside of our cell, but just know that with the sphingosine, you can see it's a monomer or monochain fatty acid tail. So it could be a precursor to cholesterol, for instance, or it could be a precursor to many other types of things. But the four main classification are these four. Now, how do they get their names? Again, you take a look at the functional group. So for those who are in organic chemistry or have taken a nursing chemistry or some sort of biochemistry, you'll kind of recognize these groups where we have an ethane group with an amine group right here. So that's ethanolamine right there. We can also have a serine, which is your amino acid serine right here, bounded to that phosphate group. We already saw phosphatidylcholine, which is an amine group, right? Very similar to phosphatidylethanolamine, except we're replacing all the hydrogens with the methane group, and then sphingomyelin, which is again the exact same as phosphatidylcholine, with the exception of the fatty acid tail and chain. So this one's a fatty acid chain. This one's a fatty acid tail. The difference is where the carbonyl slash carboxylic group is. This one doesn't have a C double bond O, so it's a fatty chain, versus a fatty acid, the acid representing the carboxylic acid, the COOH behavior, which we don't have anymore. Okay. So again, the shape of cholesterol, just recognize the shape. It's a four-membered ring. It is a single, very similar to a single chain fatty acid there will be a polar region and a non-polar region right here. Again, that plays a big role. Now, if we take a look at this next slide right here, we can see that a lot of the times we're going to have cholesterol embedded inside the phospholipid bilayer. Now, again, cholesterol is one of those weird things where depending on the concentration, it either makes it more fluid or less fluid. But I don't want you guys to get too caught up on that. Just know that cholesterol is there to kind of inject the instability of stacking structures. But in small concentrations or in some concentrations, we do see the cholesterol stiffened region. So what that means is because of their polar and non-polar regions, the fatty acid tails of phospholipids is going to heavily interact with the cholesterol ring right here, as well as the phosphate head interacting with the OH group of the cholesterol. Because of this, it's going to create regions of magnetic attraction or electro non-covalent interaction where these phospholipids will stick to this cholesterol in a clump. Now, again, depending on how many cholesterol we have versus how little, that again plays a role in the fluid-like region. But just remember, the kink of the tail or the bend of that fatty acid tail is what adds to the fluidity. It prevents the stacking of completely saturated fats, uh, which again makes it more liquid-like. Okay. Again, phospholipids spontaneously will form uh, bilayers when they can. But in the event that something is dissolved in water, remember, water has a bipolar type of behavior. It has the properties of cohesion, adhesion through the interaction of hydrogen bonds, and it can create a hydration shell. Now, it's the hydration shell from chapter 2, we've talked about this already, that prevents the ionic characteristics, for instance, or other soluble molecules from staying a solid within the water. Reason being is because depending on the charge, whether it's partial, positive, or negative, water is going to surround that individual molecule and split it off from its crystalline structure. Just think about it. If you guys were going into a concert as a group, say that five of you guys decide to go to, an Ed Sheeran concert. I don't know if I'm pronouncing his name right, but bear with my analogy here. Say that you go to his concert, but then a huge crowd came in because, the singer was right in front of you guys. And then all of a sudden, everyone started surrounding everybody else in your group so that you guys all split apart. That's essentially what water molecules are doing in these types of solution. They're surrounding the acetone molecule, for instance, completely encapsulating it in water, splitting apart from the rest of the acetone molecules. And same thing with methyl propane. Okay. So methylpropylene, again, depending on the circumstance, there are certain cases where we do create hydration shells around nonpolar molecule. Methylpropane being a nonpolar molecule, you can see that water formed a shell around it. Now, this is typically not the case. But in the event of a property called emulsification, which is salad dressing, you know how you buy salad dressing at the market, it's separated into two layers and you have to shake it up and it creates one nice homogeneous layer. Very similar. That is essentially what's happening when you shake oil and vinegar or oil and water together. Water surrounds the oil in form of a bubble and you have an emulsified product. But again, in a cellular content, that's not necessarily the case. If we were to pour oil, just regular oil on top of water, it's going to form layers where one layer is the water, which is on the bottom, and the other layer is the oils. Say that you put olive oil in a mason jar full of water, you can see that. Now, here's the question though. Why is it that when we have a fatty acid molecule that only has one tail, Does it form a little ball shape? But when you have two tails, it forms a layered shape. And really kind of just think about it. Look at the shape that it makes. If you were to draw this out, right? If you were to draw this out, a single tail versus double tail, you can see that this single tail forms a cone-like geometry. And it's this cone-like geometry that allows us to create these structures, these ball-like structure called micelles. And inside the micelle, it will carry something that's non-polar where it can be transported. Now, if it was a double tail like this, right? And we try to form a little ball shape, it's not going to work too well, right? How's that going to work? So let's try to draw a little ball shape here where I'm kind of trying to bending it in, right? You can see that there's a lot of empty spaces around. I can't really fully draw a nice little ball, and it just gets very sterically hindered. I mean, try to try to create a little ball with soda cans. Like buy a pack of Coke, a Coca-Cola soda at home. Maybe you have a six pack at home. Try to morph it into a sphere shape. It's very hard to do. Versus if you have a whole bunch of like drumsticks, the ice cream drumstick, you can kind of form it into a little nice, little geometrical sphere. And so that's the case. So in the event that we kind of damage our lipid bilayer, say that we poke a hole right here, what's eventually going to happen is that it's going to flatten out into a bilayer shape. It won't form little curves right here because spherically, cylindrically, it doesn't geometrically make sense to. It is very energy inefficient, and due to the polar non-polar region, it's very energetically unfavorable to keep it in a semi-sphere shape. Okay. So the next thing, again, we got to talk about is understand that the lipid bilayer is fluid. I just said this a couple slides ago. It's moving constantly back and forth through lateral diffusion. Okay. It's also rotating. The phospholipids can rotate, just like if you have Coca-Cola at home, cans of Coca-Cola, you're able to turn each can, no problem. All right. There's really no issue with the spinning of the can or the movement of the can from side to side. However, if you're trying to flip one of the phospholipid from one orientation to the other layer, it's very hard to do. Again, you have to be able to push the phosphate head, which is polar, through the fatty acid region and flip it to the other side. It's incredibly, incredibly hard to do. Now, it isn't impossible. It's not impossible. It's highly unlikely without the aid of a flipase or a fatty acid translocator protein. Flipase is more so what we would be considering. Okay. Now, just know that, for these movements, it happens in microseconds, nanoseconds, even picoseconds sometimes. It's just constantly in motion. Okay. Now, if we take a look right here, again, fluidity is based on the stacking ability of the fatty acid chains. The easier it is to stack, the more solid-like it becomes, just like butter. Butter is solid at room temperature, but if we add some unsaturated fat, remember, so the term unsaturated, and let me write it down, unsaturated by definition means you are not fully saturated. Saturated. Unsaturated represents the amount of hydrogen that is bounded to carbon, right? And if we are unsaturated, that means those carbon bonds are not fully saturated with hydrogen, which means there is a double or a triple bond somewhere in the fatty acid chain. Because of that, we have these little imperfections in the stacking right here. And if you try to stack it, it won't, because it just doesn't stack very well. It kind of topples over and then becomes more fluid-like. Versus if you have a fully saturated, completely saturated hydrocarbon chains, You can stack them. It's very easy. It's like flat wood, like flat logs. You're able to stack them very, very simply. Okay. Now, do take note that the unsaturation is forming from a cis double bond structure. So if I was to draw a cis double bond structure, and for you OChem nerds out there, this is what we're talking about. And let me wipe the screen out. For a cis double bond structure, what we have is we're going to have a carbon bounded to another carbon, which is double bounded to another carbon. And depending on where the next carbon chain is, whether it's on top or on the bottom, will determine if it's going to be cis or trans. Since we are looking at cis double bonded, we're going to look at a carbon structure that has carbon bending in the same side or to the same side. This is the cis conformation, and we can see the little bend right here. Okay. And for a trans conformation, we can form the same three bonds, except carbon is now bounded to the top. Now, unfortunately, in the trans configuration right here, when we're talking about cis or trans saturated fats, the trans fat right here is easily stackable, right? Because now you have a layer that, yes, it's a slight bend, it's a slight imperfection, but it's stackable. We can work with that because again, hydrocarbon chains are just zigzags of each other due to the tetrahedral geometry of carbon. Versus cis bonds right here, if we were to draw it in a skeletal structure, it's not going to be able to stack with the other fatty acid chain. You can see the bend is causing some sort of imperfection right here. So this, and again, let me pull my laser pointer out here. If we can see this bend right here doesn't allow the stacking of another fatty acid on top of it versus this one looks kind of straight. You're able to stack it much easier uh, than not. And this is why a lot of companies back, in the 90s, like were creating vegetable oil that was trans fat, right? Because we thought back in the day that vegetable oil was healthier. Rightfully so, because again, it's unsaturated fat. However, once we incorporated some chemical reaction where we changed the liquid nature of the unsaturated fat to a unsaturated trans fatty acid, and became solid, like I can't believe a lot butter or margarine, etc., that's where things get a little bit hairy because we actually don't have the tools in our body enzymatically to break a trans fatty acid. It actually stays with you forever in essence and gets passed down to your children. At least that is what was taught back in the day when I was first learning it. Things might have changed now. I don't know. But more you know, right? Anyways, so with that being said, again, this is just extra information, but something that should be nice to kind of take a look at. And then right here we have the lipid composition, what's contained in the lipid, depending on if it's a liver cell, red blood cell, myelin sheath in the nerve cell, mitochondrion, etc. And you guys can take a look at the differences. Now, last but not least, again, there are a lot of things inside of our membrane: cholesterol, fatty acids, even proteins. Now, excuse me. Now, before I get started talking about proteins, excuse me, and the type of interaction that proteins have in our phospholipid bilayer, just know that there is some sort of theory out today. We don't fully understand it yet, but due to the sticking behavior, you know how cholesterol, if we insert cholesterol in, it creates a stiffness to the membrane. There is some theories that states that depending on the circumstances and the function required, sometimes we create a stiff like structure called a raft inside the membrane because it is needed at that point. And what's involved in the membrane is your phospholipids, your cholesterols, as well as any surface protein or transmembrane protein that is required in that particular area. So if we take a look at this membrane, we can see that we have these transmembrane protein as well as these anchored surface proteins. And we can see a lot of other things involved, but just know the RAF protein is one of those interactive non-stationary structures that the lipid membrane can create from time to time. Now, one thing I want you guys to start taking a look at is just the labeling itself. Look at the shapes. We have hexagons here. We have the yellow circles here. And again, I'm hoping that you guys are starting to kind of get the sense of what we're trying to show. Hexagon being the sugars, yellow being the phosphate group. But you can see that a lot of your phosphates sometimes doesn't even have a phosphate group right here. This is a glycolipid right here. You have a fatty acid chain with only glycogen or glucose molecules or sugar molecules, oligosaccharides more so. You have anchor protein attached to phosphoglyc what is it? Oligophospholipid right here, right? You have transmembrane proteins that are embedded throughout. You have multi-pass transmembrane proteins. So again, this is all part of that soup-like structure. And again, pay attention to how the transmembrane protein look. Look at this. It looks like a corkscrew, right? So what structure of the protein is running through the membrane? The thing that looks like a corkscrew, for the most part, are your secondary alpha helical structures because they are the structures that can have an amphiphilic behavior, and it's the simplest structure that we can create with an amino acid chain. Now, it doesn't mean that beta sheets can't go through it. Yes, we can form different structures called a beta barrel, but that requires a multi-pass beta sheet structure versus an alpha helix. You can have a single alpha helix running through. Okay. So, asymmetry of the lipid bilayer is functionally important. Again, we don't want lipid bilayer to be completely symmetrical because, again, the environment from inside the cell and outside the cell are not the same. Okay. So, you have extracellular space. Whoops. On the outside of the cell, you might have a lot of glycolipids. And again, the glycolipids are required, right? Some of the glycolipids are used for signaling purpose. They're used for receptor purpose. They're used to hold your cytoskeleton together or hold armor, cellular wall on the cell, depending on what cell you're looking at. Now you also have a cytosol region right here where it is all negative charges. And again, it just depends on what's inside the cell. Now, in the next chapter, we will talk about, or later this chapter, sorry, we will talk about the positive and negative effect, especially when we're talking about the sodium potassium pump. But we'll talk about that when the time comes. I believe it's this chapter. Yeah, it's part of this chapter. Okay. Now, sphingolipids. Okay. Sphingolipids, pretty nice. They are essentially lipids with sugars on them. Okay. And if we take a look at the structure, you'll notice that you have galactose as one of the sugar molecules. You have another compound called nana, which is a celiac acid. But just know that these are just different types of patterns that your cells are going to recognize. And when I say patterns, I'm talking about the moieties or the motifs that are recognized by certain types of proteins and certain types of other uh, uh, cellular receptors or just other enzymes that need to kind of be like, hey, where am I going? Kind of idea right here. And so, is this going to be something where it's going to play a huge role later on in this chapter? Not necessarily, but it is good to know that, depending on the sugar molecule, we can alter the sugar molecule. So you have a glucose molecule right here. We can alter what's attached to it to create a different variant of that sugar and cause a different type of signal or a different type of receiving signal. Okay. All right. So membrane proteins. Again, Lots of different types of membrane proteins. Uh, there's just so many. The simplest one are your alpha helical transmembrane protein. So you can see that it's the alpha helical shape right here. But also notice that sometimes we want the protein to be as stable as possible within the membrane. So you might have a region of the amino acid where it covalently binds to a fatty acid, or it covalently binds to a phospholipid, or covalently binds to a cholesterol to anchor itself inside the membrane so it doesn't float away, for instance. You also have different types of transmembrane protein that is multi-pass transmembrane protein, where you can see that there's three alpha helicals that form a function. You can have one that's predominantly only on one side. You can have one that's on the other side. And this is what I mean in number five, where you have a beta barrel structure. Now, this beta barrel most likely is going to be a pore-like structure. Same thing with this alpha helical. You have multi alpha helicals right here. Chances are that's going to form a specific motif that will be very selective on what it allows to pass through it. Generally, these are going to be your facilitator proteins or your translocator proteins or your transporter proteins, etc. Pore proteins. Um, and it just depends on what is attached to it, what it receives, if it's phosphorylated, is it energy activated through ATP or GTP signaling? Okay. Now you also have these other proteins right here where they are peripheral proteins. But these peripheral proteins right here are going to be embedded on one side. Okay. Now this alpha helical is going to also be what do you call it? Amphiphilic, because again, one side has to be hydrophobic and the other side has to be hydrophilic if it wants to be embedded inside the fatty acid chain or fatty acid layer. And there's several others where you're going to have different types of covalent bound again to a phosphate head with oligosaccharides on it, etc. Or you can form a dimer with another transmembrane protein to create a specific function. So again, proteins can be released depending on what is attached to it. Again, they have different types of linkages. We have amide linkages where we're connecting uh, one of these proteins right here through the amine positioning on this fatty acid group. Okay. These are all, again, all covalently bounded. So to kind of remove this will create quite a bit of energy. We also have proteins that can bind using a thioester linkage. We have other proteins that, again, uses different types of bounding depending on what is attached to it. Okay, it could be a direct sulfur group on a cysteine, or it could be on a prenyl group where a sulfur and a cysteine is found. You can also have different types of anchors. Right here, you have the myristoyl anchor, the palmitoyl anchor, and the farnesyl anchor. Okay. But these are all different ways that proteins can either be attached or released depending. Okay. Again, with any type of cleaving, it will require some sort of hydrolysis type of reaction and some sort of enzyme to aid in that type of reaction. Okay. So if we take a look right here on our next list, we have the alpha E helix crossing the plasma membrane of the level structure. But we see that the R groups, a lot of the R groups, you have your glycine, you have your phenylalanine, you have your tyrosine, you might have your serine. They're all non-polar molecules. Again, that makes sense. We want them to be found in the fatty acid layer. Now, the question is, how do we tell if we're to kind of analyze an amino acid chain in a primary structure, how can we tell which regions are hydrophobic and which regions are hydrophilic? And if so, is that going to kind of represent a transmembrane protein? Well, in all honesty, yeah, there is a tool that we have, and it's this next slide right here. Now, this tool is what we call a hydropathy index, where we can pretty much identify the overall behavior or the overall charge um, of whether something is hydrophobic or hydrophilic. Now, we take a look at the hydropathy index right here, we can see that amino acids from 0 through 50 is a negative charge, meaning that you don't need any energy to interact with this region of the amino acid in water. So if you don't need any energy to interact with this region in water, chances are it's going to be polar, which makes sense, right? Because, if you're going to put something in water, there's not going to be any resistance to it. So no energy spent to interact with water, if that makes any sense. However, once it goes above the zero charge and goes into the positive charge, this represents an area where the amino acid requires energy to interact with water. Because again, with the repulsion effect, right, from a hydrophobicity to a hydrophilicity, having water try to interact with this fatty acid, it's going to take a lot of energy just to kind of make an emulsified structure or have it interact in general. And because of this, you need to spend energy. And that's what this positive charge means. It means that you're spending energy to interact with this. So if that's the case, from amino acid 50 to amino acid 70, this is going to represent a hydrophobic region because it requires a lot of energy to interact with water. And then after that, you have another 25 amino acids here that shows a negative interaction with water, which is meaning it doesn't require any energy to interact with water, representing a hydrophilic region. So we can see that on this amino acid chain, and let me get my pen out here, on this amino acid chain, we have one transmembrane region because it's a hydrophobic region. It's going to pass through the membrane at this specific area. And then that's pretty much it. So this hydropathy index shows how many regions on an amino acid chain there is that passes through a membrane. If we look at the next one, we can take a look that on this next one right here, we can see that there are 1, 2, 3, 4, 5, 6, 7 regions. Now, it's a little wonky with the way they drew this. I don't like how they didn't include these regions here, but they're essentially taking the highest peaked regions uh, and labeling that as the hydrophobic areas. Okay. So it would be this as 1, and then this big one is 2. This one is 3. That makes sense. This one is 4. This one to this one should be 5. This one to this one should be 6. And then this one to this one is 7. So it's still 7, but they're trying to make it really clear cut for you guys so that you guys don't kind of misinterpret it. But these are 7 regions. And if we have 7 hydrophobic regions, chances are this is a multi-pass transmembrane protein. And if we take a look, it is a multi-pass transmembrane protein. This is one of the examples where we can see that 2 of the alpha helix is not a transmembrane helix, but it is a region found within the pore of the helical structure where it can be extremely selective to what passes through it. So most likely these alpha helix are not hydrophobic. Remember, if we take a look from the outside, you have 1, 2, 3, 4, 5 alpha helices from the top side view. So if we took this membrane right here and we look at from top down, we can see that these transmembrane protein form this little circular structure, this little 5-ring pore right here. And inside the pore, we might have your green or your blue. And let's just use a different color to kind of show. You might have a blue alpha helix inside here, right? Now, this alpha helix, because it looks like this is a pore structure, remember the alpha helix in the red that I drew has to be hydrophobic. It's interacting with your fatty acid tails. But inside the pore, because it's a pore and stuff is flowing through that pore, what is the stuff that flows through the pore? It's water. It's something that is soluble in water. So the inside of this area right here has to be hydrophilic. Now, if it's small enough, it doesn't have to be. If it only allows one atom to go through, then it doesn't have to be hydrophilic. Okay. But for the most part, it will be hydrophilic. And the alpha helix represented in the red here and the blue here, or green, I can't tell what color that uh, is also hydrophilic. And this is going to cause some sort of selectivity. Okay. Now we have a lipid bilayer. These are your beta barrels. Again, there can be stuff inside these beta barrels. Later on, we'll talk more about that, where there might be some mesh-like structure that adds like a filter structure, especially when we're talking about nuclear pores. Okay, that's going to filter out a lot of things that can pass through it. Okay, but take a look at how many beta sheets you need to create a beta barrel. You need a minimum of at least 10. Versus Alpha Helix, you just need 4 or 5. So, okay. Now, next thing, glycolipids. Your proteins need some sort of, well, it doesn't need it, but it is altered quite often on a membrane. And most of the time, yeah, you're going to have multiple sugars found on these proteins. Now, you guys have probably cooked steak before. And if you do, you'll notice that if you leave your steak alone, say that you have a hot skillet, you put a steak on, the common practice or the best practice of having a nice sear on the steak where it's a nice brown crust is what we call um, caramelizing the steak. Now, we only ever use the word caramelizing when we're trying to make caramel. And caramel or caramel, depending, is the process of heating sugar and cooking it. Now, why are we saying that for steaks? Well, the main reason is because steaks, as portrayed in this slide, in terms of amino acids and proteins, have sugars found on them. And these sugar residues are used in signaling, they're used in receptors, they're used to create protection from the outside environment. Hence why, with a lot of sugar molecules, if you take a look at steak, most of the time it is very smooth, right? The meat is very smooth, it's very silky, slick. Now, fat has some things to do with it, but mostly it is just very good. Whenever you get a burn, what did they say? Use aloe vera, which has a lot of sugars in it, to repair the burn marks. Reason being is because, again, sugar molecules are needed to create a layer of coating, a layer of slick-like protection. Now, these sugar molecules also interact with your cytoskeleton, with the overall structure of the cell itself. And so we see a lot of these on the surface of the membrane. And if you take a look at image A right here, it shows that in the electron micrograph. Okay. Now, in terms of detergents, all I really want you guys to know is that detergents are a amphiphilic molecule. They are monomeric, but they can form complex structures like micelles. And if we want to destroy a membrane, or if we want to kind of get rid of impurities, we can use the amphiphilic behavior of these micelles or of these proteins or these detergents, sorry, to kind of surround a protein or surround lipids or surround grease, impurities, etc., forming their own little micelle bubble, like hydration shells, but it's little micelle shells, and be able to clean them off. And this is what we do when we are developing soaps in our world. They are natural, they are detergents, and they act like natural detergents. Okay. All right. Now, we are now entering the next chapter, chapter 11, right here, where we are going to be talking about different types of proteins, channel proteins, glycolipids, and um, other pieces of information. So bear with me. This slide is talking about the sodium potassium pump and the involvement of detergents. All you really need to know about this slide is that in order to isolate the sodium potassium pump for us to study it, we use detergents to kind of remove them from a cell membrane, incorporate them into micelles, and then use that to study them. Okay. Again, not gonna be too involved with this slide, but it is something that you guys need to know slightly when we are in the process of purifying our protein of interest. So this is like a review of chapter 8. What we can do instead of using our column chromatography, we can use detergents to kind of purify our proteins. Okay. All right. So this is a great stopping point. If you guys need to stop right here, I recommend it. But in chapter 11, what we will talk about is the sodium potassium pump. Now, this is a 40 something minute video. I will be editing this, so hopefully it gets down to around 30 minutes. You guys will see the final product should be around 35 minutes. But I'm hoping that this first segment is good. If you have any questions, let me know. But this PowerPoint will be a multi-video PowerPoint, so I will separate them by chapter so that you guys have an idea of how to focus your study. So I'm going to stop it here, and I'll see you guys in the chapter 11 uh, video. Okay, so see you guys in the next video.